happens first, generally people present in the clinic with memory deficits. These are usually spatial memory, so you'll forget where you parked the car, or where your keys are, or where you put something, or how to get somewhere. But as the disease spreads through the brain, more parts of the brain become affected and more it progresses. After just memory deficits, we usually start to see impairments of other thinking and cognitive domains like language deficits, planning deficits. But during Alzheimer's disease, the brain goes wrong in lots of different ways. You can see here a control brain, and next to it, the brain of an Alzheimer's disease patient. And you'll be able to tell, even without a PhD in neuroscience, that there's a big difference between these two. We have aggregates of proteins called plaques, and they're in red here, and tangles in green here. And these only accumulate in patients with Alzheimer's disease to a great extent. So you gain and lose synapses through your whole life. And these in Alzheimer's disease are hit hard and they're hit first. Dying of the synapses correlates most strongly of all the things I told you go wrong in the brain with dementia. So the loss of these little synapses seems to be driving the cognitive decline and the dementia. So some people, when they die, especially people who've had dementia, agree to give their brains to us, or tiny, tiny pieces of their brain, for research. So what we have here that you can probably just see is a whole bank full of drawers, and each of these drawers contains these tiny little blocks. Each of these tiny little blocks contains an even tinier piece of human brain right at the bottom. So this is the hippocampus of a person who did not have Alzheimer's disease. Remember, the hippocampus is really important for learning and memory. The blue are the neuronal nuclei in nice little lines, and out here, this is the neocortex hippocampus. And I want you to notice how big and healthy this hippocampus looks. And now what I'll do is change the slide. So now this is the hippocampus of someone who had Alzheimer's disease before they died. And I want you to notice a couple of things. One, it's smaller because it's shrunk and a bunch of the neurons have died. And two, see those dark, almost black spots? Those are plaques. If we go out to the cortex, the plaques are even more prominent. Look at that. So the brain is completely full of this plaque protein. It's also full of the tangle protein, and the immune system that occurs in the brain called microglia and astrocytes also flare up, so you have a bunch of inflammation in the brain, so lots of things going wrong. The brain is shrinking, and all these pathologies are accumulating. We've been able to take this kind of picture for over 100 years, since Alzheimer discovered the disease in 1906. But what I'll show you next is the really thin sections we were just taking, and how we look at the individual synaptic connections, and how these pathologies affect those synapses. I'll show you now how we cut the sections, and then we'll go and I'll show you how we look at them on the microscope to see what's changed.
At the autopsy, the coroner saves small samples from each area of the brain and puts them into glass vials, and are then passed on to us for experimentation. First, cut the provided tissue block in half. We then take the other half of the tissue and carefully cut it into 1 mm by 1 mm by 5 mm blocks. These small blocks of tissue are then placed into a solution of paraformaldehyde, which helps preserve the tissue structure as well as stopping the breakdown of proteins. After being incubated in paraformaldehyde, the tissue is then dropped into a resin and labeled. The tissue and resin are then baked. This results in a hard block with tissue embedded in it. We then need to cut the tissue into 70 nanometer sections. To do this, we mount the block containing tissue into the arm of an ultramicrotome. We then secure a boat which houses an ultrafine diamond knife on one side. Into this boat we place a glass slide and then fill the boat up with water. Once we have cut 20 to 40 of these sections, all joined together in what we call a ribbon. Hydrophobic pen is then used to draw around the ribbon, which allows us to apply liquid directly to the ribbon without it all falling off the slide.
The next one. So these are serial sections through the brain, and these little white dots are the neuronal nuclei. What we use this for is to make a map, so we can find the same spot on two sections in a row. For example, this is a little blood vessel here in this bit of human brain, and you can see all these little endothelial cells, the nuclei of the cells that surround the blood vessel, and you can see them on this section. And if we zoom over to the next section, we can find exactly the same pattern of nuclei. So we use this as a roadmap to find the same spot, so we can take a 3D stack all the way through the brain. Then we move up to a much higher resolution objective where we can see things much better, and we take a picture in all the different channels that we've stained. So if I show them to you one at a time, these are the nuclei. So you can see that's the nucleolus. This is where the DNA is of the neurons. This is now the plaque. So these are the things that were labeled brown when we looked on the thicker section. Now we can see them in much more detail labeled in, now it's red, I'll show you, but right now it's just white. This is ApoE, so this is a protein that's involved in risk for Alzheimer's disease, and we're trying to find out how it affects these little synaptic connections. And these tiny dots are the synapses. So these are the connections between the brain cells that we're studying. And what we do when we look at them all together is we can say, for this little purple synapse, does it have any of this red plaque protein? And we can zoom in and we can find examples of synapses that have little purple synapses that have either green ApoE or red A beta or both. So they have pathological proteins at the synapse. We can go through 3 dimensions. So if we move through the stack, you can see we don't just have one single section, we have about 30. And once we figure out what's going wrong in the synapses, what we try and do is go back to our models and remove these pathological proteins and see if the models get better, see if the synapses come back in our models. We don't have any treatments that are very effective. We have 5 or 6 approved drugs for dementia, and they help the symptoms at certain stages of the disease, but they don't stop the relentless progression of the disease through the brain, and they don't help you get any better.